Hello everyone, my name is Rebecca Harris, and this is my presentation for my Descriptive Epidemiology project on Nipah virus. In this presentation, I will introduce Nipah virus and its transmission as the main problem of focus, as well as how it is transmitted among southern and southeastern Asian countries, hypotheses for prevention, and suggestions for the future. Let's get started. First, the definition of the problem. Nipah virus, pronounced Nipah, yes, I initially pronounced it wrong too, is a rare, contagious, and potentially devastating disease found in southern and southeastern Asia. It is highly contagious and has the potential to become a devastating epidemic. According to the World Health Organization, NIV, which is short for Nipah virus, is one of the pathogens on the priority list most likely to cause outbreaks needing urgent research and development action. I've displayed two separate views of Nipah virus. The first is from a microscope, and the second is a view of the molecular structure of the virus. There are 6 main proteins in Nipah virus, seen here as the G, F, M, N, P, and L proteins. These work together at the molecular level to assist the virus in invading healthy cells and spreading the virus throughout the body. Next, we will discuss the description of the agent and the condition. So while this little guy, this little bat in the corner, looks pretty cute, he's actually one of the species of bats that hosts the NIV virus. NIV is defined as a zoonotic disease, which means it comes from animals. NIV is part of the Paramyxoviridae family in Henipavirus genus. From a molecular perspective, it is an enveloped pleomorphic virus with a genome consisting of a non-segmented, negative-sense, single-stranded RNA. In simpler terms, it's a very complex virus that can do a lot of damage. NIV's transmission has been found in many mammals, including pigs, dogs, horses, and even humans. It is primarily passed along species through contact with body fluids, such as blood, saliva, and urine. In some Asian countries, NIV has even been found in date fruits and their trees. We will discuss that later in the presentation. The incubation of NIV ranges from 4 to 14 days in humans. On the next slide, we will observe a diagram of the transmission of NIV. So this graphic may be a little overwhelming at first, but this is a depiction of the transmission of NIV between species. Starting here at number 1, fruit bats infected with NIV are going to feed on the date palm sap. This sap is then harvested by humans, which then infects humans when they're in contact with the sap, which contains the saliva from the infected bats. Looking at number 4, in a similar scenario, bats feed on fruits. Their infected saliva enters the fruit, but instead of humans directly coming into contact with NIV, pigs eat the fruit. When pigs are then taken care of on farms or sent to slaughterhouses and or consumed, NIV is transmitted back to humans. Finally, looking at number 7, human-to-human transmission of NIV is possible when humans are in contact with contaminated body fluids of other infected people. This is most common in medical centers as medical staff attend to the sick, and in villages where small communities frequently interact with each other. Symptoms of NIV include fever, headaches, myalgia, vomiting, and sore throat. In severe cases, NIV can cause mental confusion, disorientation, and coma within 1 to 2 days. These extreme cases can also exhibit encephalitis, which is the swelling of the brain, seizures, septicemia, impairment of the renal system, and bleeding in the gastrointestinal tract. Down here in the corner, I've included a picture of a brain scan of a person who has had NIV. Unfortunately, they did die from the disease, but they did donate their body to science so that way researchers can learn more about the disease. And that is a scan of the effects of NIV on the brain. Unfortunately, there is currently not a safe and effective vaccine for use in humans for NIV. Luckily, there are a couple of contenders in production. Until an improved vaccine is created, treatment for NIV primarily relies on palliative care, which means keeping the patient comfortable, such as keeping them hydrated, fed, isolated from others, etc. The best methods to prevent NIV transmission is to practice standard precautions such as hand hygiene, washing your hands, using hand sanitizer, and in medical environments, using personal protective equipment, or PPE for short, such as gloves, masks, gowns, and other similar devices. Next, we will talk about the distribution of the health problem. As mentioned previously, NIV is primarily in southern and southeastern Asian countries, which is seen here on the red zone on the map. One of the complexities of NIV is that it presents a different mode of transmission in each country. We will primarily focus on 5 different countries: Malaysia, where NIV was discovered, Singapore, Bangladesh, India, and the Philippines. The first case of Malaysian NIV was in 1998. Originally, scientists thought it was Japanese encephalitis virus. This was actually a fatal mistake, as people in the country took preventative measures for Japanese encephalitis, which are unfortunately different prevention measures than are needed to address NIV. In Malaysia, it is common practice to raise pigs, which, as we learned from the previous slides, carry the NIV virus. When farmers came into contact with the bodily fluids of pigs, they contracted NIV. These cases later progressed into Singapore, as the pigs from Malaysian farms were transported to Singapore to the slaughterhouses. Moving on to Bangladesh. Unlike Malaysia and Singapore, pigs were not involved in this route of transmission. After some extensive research, scientists discovered that the sap in date fruit trees was a reservoir for NIV. In Bangladesh, it is common practice to harvest date fruits and the sap from their trees. In some places in Bangladesh, this food is actually considered a delicacy. As the local bats, which we learned on the previous slide are also carriers of NIV, drank the sap or bit into the fruits, their saliva entered the crop. This contamination was later distributed to humans as they too consumed the sap. Since that time, NIV has become known as a seasonal illness, with most cases arising between December and May, which happens to coincide with the harvesting of date fruit sap. This pattern has earned the area the name Nipah Belt due to the regularity of cases. As of 2023, there are currently 11 cases and unfortunately 8 reported deaths from NIV. Moving on to India, there have been 3 different epidemics or outbreaks of NIV. The first in Kerala, which is shown here with the red star, another in Siliguri, and another in Immaculum. Researchers were initially confused as to the source of NIV since there were no pigs reported of having NIV, similar to Malaysian NIV. Similar to Bangladesh, however, scientists studied bats and found that some of the bats in the area did test positive for NIV. Despite this discovery, researchers maintain that there still needs to be more intensive studies to fully understand NIV in India. And finally, we'll take a look at the Philippines. Unlike the previous cases, Philippine NIV was not found in pigs or bats. Actually, in the Philippines, it is common practice to raise, slaughter, and later eat horses. When villagers came into contact with body fluids of infected horses, they became infected just as the farmers in Malaysia were infected by pigs. Let's talk statistics about NIV. I have broken it up into two different columns, one for morbidity, which is the rate of disease in an area, in this case, which is the number of cases in each area, and mortality, which is the number of deaths. Each of these categories are further broken down by country. Starting in Malaysia, there have been a total of 265 cases with a 41% mortality rate. In Singapore, there have been a total of 11 cases with a 10% mortality rate. In Bangladesh, over the course of 3 epidemics, there have been 261 cases with a 69% mortality rate during the first epidemic, 83% mortality rate in the second, and 77% mortality rate in the third epidemic. In India, over the course of their 3 epidemics, there have been 85 cases reported with a disturbing 90% mortality rate. And finally, in the Philippines, there have been 17 cases with an 80% mortality rate. After researching NIV, there are 3 hypotheses that can be concluded. First, if the climate continues to change, then NIV cases will increase. NIV needs to survive in a suitable host, whether it is in pigs, dogs, bats, horses, or date fruits. If the Earth's temperature continues to rise as projected, the habitat for these hosts will expand. This will increase the chances of NIV spreading to other countries. The second hypothesis. If the methods of transmission of NIV are addressed in time, then cases will decrease. As mentioned previously, the mode of transmission of NIV differs in each country. In order to prevent the further spread of disease, scientists need to pinpoint the exact method of transmission. Since they know that NIV can survive in mammals as well as sap from date fruit trees, testing of these sites is crucial to catching the disease before it spreads uncontrollably. Finally, the third hypothesis. If people are educated about NIV, then they will learn to prevent infection. NIV is not a commonly discussed disease here in the United States, but it has devastating effects on Asian populations. If those populations are educated about the spread of NIV, then they will be more likely to check their crops for infection and catch the virus before it spreads. There are two principal gaps in knowledge. The first, despite its presence in Asian countries, there's very little knowledge about NIV and an effective vaccine. Unfortunately, for human trials to begin on an NIV vaccine, more people would have to get sick. There is a vaccine in production, but again, they can't conduct human trials because there are not enough cases. Also, there is very little knowledge about how the virus would survive in other areas with a comparable climate. Since the virus is mainly secluded to Asia, it is difficult to make conclusions about the virus if it were to progress to other continents. Here are a couple of suggestions for further epidemiologic research. Epidemiologists need to learn more and study NIV, its modes of transmission, and the lack of a vaccine, which all of those combined provide NIV with the capability to become a pandemic. Scientists can use the knowledge obtained from previous cases to determine effective preventative measures for the future. Some people in the aforementioned Asian countries have agreed to donate their bodies to research, which is helping scientists learn about the effects of NIV on the human body. Another avenue for further epidemiological research is to monitor bat colonies over time. If these colonies choose to migrate, it is a safe assumption that NIV will spread to other areas as well. NIV is a rare, contagious, and potentially world-threatening disease. If epidemiologists can learn more about the virus, its transmission, and possible progression to other areas, hopefully a vaccine can be created and we can be prepared as a global community to address this public health issue. Thank you all very much for watching. Here are some questions for you to consider following my presentation. And finally, on the last slide, I have my references and resources. Thank you all for listening.